All right, hello. Today we're going to continue our conversation about the adaptive immune system. And today we're going to talk about the different types of cells involved with our adaptive defenses. So those would be our B and T cells, and also a category of cells called antigen-presenting cells. So let's start off with talking about the B and T cells and where they come from. So, where do they come from? Where do all of our blood cells come from? Hopefully, you're saying red bone marrow at home, because that is the correct answer. And then they have to mature, and they mature in primary lymphoid organs. So, T cells mature in the thymus, and B cells mature in the red bone marrow. And let's talk about the maturation process in a little bit more detail. So, when the lymphocytes are maturing, they need to become immunocompetent, which means they're able to bind to and recognize one specific antigen. And then they also have to become self tolerant, which means they do not bind to self antigens. If a lymphocyte recognizes one of our own cells as something foreign, that could trigger an autoimmune response, or when the immune system attacks itself. This process shows how they mature, so how they become immunocompetent and self-tolerant. And this is specifically called T cell education, but the process in B cells is similar. So there are two steps, starting with positive selection. The T cell needs to be able to recognize the MHC protein. If you remember from our other conversation, the MHC protein is on the surface of the cell and it contains a little peptide. So the MHC part is like this purple rectangle, and then the orange thing is the self antigen in this case. So here's a T cell, and we can see that it has like a triangle right there when it needs to have a square. This cell is not able to recognize the MHC thing, so we kill it. Down here, look at this T cell. They are binding perfectly, which means that it's able to recognize it. And all of the T cells that are able to recognize the MHC protein are able to advance to step number 2. Step number 2 is negative selection, where we want to make sure that they do not recognize the self antigens. So here we have them binding to the MHC, which is good, except look at this T cell. It bound tightly to that orange self-antigen. That's bad. This cell dies. So here we have a T cell that is binding to the MHC, but it's not binding to the antigen. That little gap right there symbolizes this. So it's not binding to the self-antigen, and that means this one gets to keep living. Could you guys imagine if I graded you like this, where the first time you make a mistake, you actually die? I guess it works for T cells because if our immune system has a mistake, we do die. All right, we talked about how they originate. We just talked about how they mature with our T cell education and positive and negative selection. The next step is that the mature cells are going to leave the primary lymphoid organs and travel to the secondary lymphoid organs. So that would be our lymph nodes, our spleen, or anywhere that we have mold, like the appendix or Peyer's patches. Once they're in a secondary lymphoid organ, they're going to actually encounter an antigen. So we call lymphocytes naive if they haven't met an antigen yet. And then the last step is after they meet an antigen, that binding with the antigen activates them and they start multiplying. Here is a summary of pretty much all of that in diagram form. So they originate in the red bone marrow, they mature in the thymus and the red bone marrow. Then they leave these primary lymphoid organs and travel to the secondary lymphoid organs like a lymph node. And this is where they could actually encounter an antigen and then become activated. And down here, being activated means the cell is going to divide. And it's going to divide into effector cells and memory cells. Effector cells are able to kind of actively fight off things invading our immune system. Memory cells remember the pathogen so that the next time it tries to attack us, we already know how to defend it. Do lymphocytes become immunocompetent before or after meeting the antigens they attack? Hopefully you said before, because becoming immunocompetent, that's part of the maturing stage, and that has to happen before they're able to leave. So since that happens before, that tells us something kind of interesting. That tells us that our genes determine which foreign substances our immune system will be able to recognize and resist. So it's not about whatever we encounter in our environment. Our genes actually specify what we're going to be able to fight off. Only some of the antigens our lymphocytes are programmed to resist will ever invade our bodies, so only some of our immunocompetent cells are used. The other ones are just kind of chilling there, waiting for something to happen. There are 1 billion different antigen receptors, like different antigenic determinants, and there are only 20,000 genes in the human genome. So how can 20,000 genes be able to code for 1 billion antigen receptors? The answer is something pretty cool, somatic recombination. So I have a Lego analogy here. This big tub of Legos is supposed to represent the whole human genome, like all 20,000 genes. And then I've taken out a small pile of them. These are the genes that specifically code for the antigen receptor genes. And then what happens is we can take these antigen receptor genes and we can combine them in different ways to express different proteins. Like here, I've made this white, blue, and green protein. And here is like an even taller white, blue, green protein. Here is a yellow, orange protein. So this is what I mean by somatic recombination, that the genes can be reshuffled into different orders, and some of them can be used at different times. And that allows just a subset of these 20,000 genes to code for 1 billion different receptors. Pretty cool. So that was us talking about the B and T cells. Finally, we're going to talk about antigen-presenting cells. The function of an antigen-presenting cell is to engulf an antigen, so it like eats it, and then it kind of regurgitates a little bit of it and it displays it on its surface. So antigen-presenting cells literally present antigens. Here we can see this little antigen-presenting cell, and he's holding this antigen and he is showing it to a T cell. So B cells and T cells need to have antigens presented to them so that they can know how to recognize them. There are a couple different categories of antigen presenting cells. One of them is dendritic cells. And if you can think all the way back to like August, our second unit about the integumentary system, we mentioned dendritic cells. They kind of look like little spiders. This is an image of one. I know that doesn't look like a spider, but when I say spider, I'm talking about like the long, wispy extensions. Maybe octopus would be a better analogy. Anyway, they are able to move through the layers of our skin, and they have these long processes, and they literally like feel all of our cells, and then when they feel something foreign, they eat it, and then they're going to travel to a lymph node where they'll present the antigen to T cells. So dendritic cells are technically part of innate immunity, but they form this really helpful link between innate and adaptive immunity by presenting the antigens. And also, I want to emphasize the structure-function relationship. The structure is having these long wisp-like processes, and then the function of that is it makes it really good at being able to find antigens. Macrophages are also antigen-presenting cells. So they are able to engulf the antigens and then present them to be able to activate T cells, or sometimes to even activate themselves to become even more voracious macrophages. And B cells can also function as antigen-presenting cells. They do not activate naive T cells like the other two categories do. Instead, they present antigens to helper T cells in order to activate themselves. So, dendritic cells and macrophages activate T cells, B lymphocytes present to helper T cells to be able to activate themselves. If that makes no sense right now, don't worry about it because we're gonna talk about that in detail in our next few discussions. Just kind of big picture, these cells are able to consume and then present antigens. The first like 20 seconds of this is a pretty good video of a cell engulfing and then presenting an antigen. And then
Activation of the
I don't want to play that right now. This is a really cool video. It's like, I want to say like 8 minutes long, but this shows the dendritic cells, and it also shows just like a really nice review of innate immunity in the skin. So it's worth a watch if you have time. Thank you.